This is Bio 181, and this video is for chapter 6, and it's all about enzymes. So the key concepts are going to be all of the characteristics of an enzyme. But first, I want to go back and define enzyme again. This is a biological catalyst. Biological catalyst. And a catalyst speeds up the rate of a reaction. L-Y-S-T. Okay. So it's something that speeds up the rate of a reaction. Speeds up reaction. And it can make it go from happening maybe like 2 times a year to happening a million times a second. No, not, well, maybe a minute. Okay. So it's basically turning it on from 0 to a million. So it's kind of like making the reaction happen, right? It's speeding it up, but from a very slow rate to something that's actually going to be functional. So we're going to go through several different characteristics that you need to know. So first of all, enzymes lower the reactivation energy of reactions. So if you have a reaction here, okay, this is our energy. And again, we can represent this with G, which is our Gibbs free energy. So if you have a reaction and you start off here with low energy and you end up with high energy, and here's your time. Okay. So here's our reaction that's uncatalyzed. So say it goes from low and then takes a lot of activation energy and you end up with your products over here, right? So again, this is your reactant and this is your product. So this energy here, okay, to go from the reactant to the product, that's our activation energy. It's the energy that's required for this reaction to occur. Energy. Now, if you have a catalyst present, so let's see, what color should I use? So maybe I will use, we're going to use red, green, okay? So if you have an enzyme present, so we'll use green here, then you're going to start off with the same amount of energy. You just don't require as much. So it's going to make it a lot less. And so it's the difference between the original energy. So this energy, right, is removed. So it lowers the activation energy. And that difference can make the reaction go from happening 2 times a year to a million times a minute. Okay? So it lowers the activation energy. And again, I just said activation energy is the energy required for the reaction to take place. So, characteristics. They bind to substrates at the active site. So enzymes are generally proteins. Proteins are complex molecules. We learned about 4 levels of structure, proteins. So they can be really large, kind of complicated molecules, right? And they have huge amount of amino acids that are part of this. There is a certain region that's called the active site. And the active site is where the substrate will bind. So a substrate, this is a new vocabulary word, substrate. This is just the word for the reactant. So it's the name of the reactant when you have an enzyme present. So a substrate is the reactant in an enzyme catalyzed reaction. So the active site is where the substrate is going to bind. So say I have a reactant over here. My reactant maybe kind of looks like this. Okay. It's going to bind right in here. So it's going to fit specifically in this place. And that's where the reaction is going to occur. Okay. Next, enzymes only catalyze specific reactions. Okay. So because if you think of that crazy shape of that enzyme, so I'm going to draw it over here again. This is just maybe a different enzyme. And maybe it has this shape. Okay. Make it kind of crazy. So here's my new enzyme. And my reactant looks like this. So here's my active site. Here's my active site. Okay, for this whole region here. Then my reactant would have a very specific shape. So my reactant, I'm going to try to mimic that, looks something like this. Right? And so this molecule is very specific shape. There are not other molecules that are going to have this shape. So this is really going to be the only molecule that fits, the only molecule that fits. So what it means is this is the, over here, this is the enzyme, this big molecule enzyme. It's only going to catalyze a reaction for something that fits it. So it's very specific. That is called specificity. So it is not a generalist. A given enzyme will not catalyze 50 different reactions. It may do one or two, but it's not going to do, so it's not a generalist. Okay. Enzymes are not consumed in a reaction, right? So the enzyme is going to be used again and again and again. So I'm going to, instead of drawing that crazy shape of the enzyme, I'm going to start using this kind of Pac-Man shape. It's simpler, right? But here's my enzyme. It's just easier to draw. And it's, you know that this is all crazy in here. This is like a very complicated molecule, okay? But I'm just simplifying it, this Pac-Man shape. So this Pac-Man is going to come along and say, here's my reactant, okay? If this is going to be moved in here, it's going to bind. The enzyme might catalyze a reaction that breaks it into pieces. So maybe I've got this piece over here, which is more narrow, and then this piece here, okay? So I've got two products, okay? 1, 2. Okay, so it broke it apart. Then as soon as that happens, the enzyme can go and let me draw this a little better. So this same enzyme, the same Pac-Man, I'll release it. It has not changed. Now, if there's another molecule over here, say you're digesting something and you have a whole bunch of food, this molecule now enters the active site, and then that gets broken up into a smaller piece. And this just happens over and over again. And it can actually catalyze thousands of reactions in a second. So it's very, very. Enzymes are sensitive to environmental factors. We talked about this already. Proteins can denature, right? Proteins denature, come apart, and they no longer function if we have a big change. I'm writing this delta, so a change in temp, or a change, this delta change in pH, which is acidity, right? This is acidity. So we know that already. Enzymes are 99% of the time proteins. So if you change the temperature or pH, a lot of times the enzymes, they may be activated by cofactors and coenzymes. Okay, so coenzymes, these are just organic molecules.
So these are things like vitamins, molecules. So vitamins that you take, cofactors are inorganic. So these are things like minerals. Right, so this might be like just iron or zinc. Oh, actually, you might not know that. So iron or zinc. So just simple compounds, whereas this might like be, like vitamin C, which is actually a complex molecule, has a lot of carbon. Remember that an organic molecule is carbon based. So what happens is you've got an enzyme here. Okay, I'm going to draw a little space in it. Here's my Pac-Man. But now the Pac-Man has a little space over here. This orientation would be off. And let me draw another one. Here's my Pac-Man. Space. Here's my active site. And this is going to be on. So this one is going to, the enzyme is going to be turned on. It's going to be turned on because it's been activated by either a cofactor, which is going to bind here, or a coenzyme. So this is an easy way mechanism to turn on or off an enzyme without just making a new enzyme every time it needs it, or breaking it down every time it doesn't. So it can just have it ready to go and it turns it on or off. Okay, so again, coenzyme, organic, vitamin, cofactor, mineral. 6, so we're almost done. They can be inhibited by competitive or allosteric inhibition. So here is your Pac-Man. This is the enzyme again, okay? And remember that this area here, mouth, if you will, this is the active site. That's where the substrate or the reactant binds. So in competitive inhibition, the active site is blocked. The active site is blocked. So something else, so say this is reactant and it wants to there to get broken down, right? What can happen is you can have an inhibitor. Green. So our inhibitor comes in here and blocks this. Right? This would be our inhibitor. And since it's in the active site, this would be a competitive inhibitor. The other type of inhibitor would be Pac-Man like this, right? And so it normally has this type of shape where the triangle would fit. But what happens is, I'm going to erase, shoot. So we've got, here's my Pac-Man. But we have an inhibitor coming along. So here, let's say this is an inhibitor. I'm just going to draw a different color, a different shape of an inhibitor. This is allosteric. Another word is non-competitive. This is going to bind here. And when it does, it's going to like push the Pac-Man shape. Change the Pac-Man shape. Okay, so it's going to bind here. Then as it does that, this is going to change. So the active site is going to be misshapen because it's going to be pushed. And then this will no longer fit. No longer fits. So this is kind of a long way of explaining the allosteric inhibitor is something that binds that is not at the active site. So not at the active site. It still changes the shape of the protein, the enzyme, in some way that it's not going to. So that shows it here. I should have just gone to this slide. Here's my Pac-Man. Right? The allosteric site over here at the bottom. You can see that once the inhibitor binds down here, it changes the shape of the active site, which was over here, this nice open shape, to something that's no longer going to allow the substrate to bind. So it blocks it. So that's inhibition. You can also have activation, which is just the opposite. So say your enzyme, the active site is misshapen, it's just hanging around in your blood, it's not actually activated, and you want to have that enzyme work, you just release a small activator into the bloodstream. It's going to change the shape of your site. If your enzyme's bloodstream, now it's going to be able to bind with all of the substrates and work. Right, the last thing, I know this was kind of a long one, I wanted to get all the enzyme together, so this is the last part, is that enzymes usually work in a pathway. So you've got enzyme 1 that's doing an initial reaction. So it's binding to that enzyme, and then you get a product. And then you have a second enzyme. The product is then going to go into that active site, and then you have another product. Again, that's going to go into a third enzyme. You have another product, and this can be like for the first stage in cellular respiration, there's 13 enzymes that are involved in the pathway. So you've got this complex pathway, and there are multiple enzyme mediated reactions. That just means reactions that are controlled by enzymes. So this results in high regulation. So we don't want to just have the body start reacting like crazy and going off. We need to keep homeostasis. We have to keep things in balance. And so if we have all of these different enzymes in place, it makes it more likely that things are going to be regulated. The feedback inhibition, so often with this type of pathway, so again, over here on the left, this is just showing a biomechanical pathway. One enzyme creating a product that goes to the next enzyme, that product goes to the next enzyme, and then you have an end product. Well, it turns out that the end product will send feedback, and maybe it will act in this case as an allosteric inhibitor. So it will shut down the reaction. So if there's a whole bunch of the end product in the area, it's going to stop the reaction. For example, if you make a whole bunch of ATP in cellular respiration, the cell will actually use that excess ATP molecule, this inhibitor, to kind of slow down that process. And just through all of our food. That's it for